disclaimer for the viewers. Let us see the characterization of liposomes. How the liposomes are characterized. There are so many characterization techniques which are available with us, which are widely used to get the idea about size and size distribution. So size and size distribution or morphology, which can be determined by using a dynamic light scattering DLS, then transmission electron microscopy and scanning electron microscopy, that is a SEM, that is size and shape, then atomic force microscopy for morphology and size, gel permeation chromatography for size distribution and homogeneity, then size exclusion chromatography in order to get the idea about size and size distribution. The transition temperature, which is obtained by using the differential scanning calorimetry, while the surface charge, it is determined by using a zeta potential and Doppler electrophoretic light scattering or the laser Doppler electrophoresis, that is LDE or the zeta sizer. The fluidity of the liposomes can be determined by using the electron paramagnetic resonance, that is EPR, or the HNMR spectroscopy and depolarization of fluorescence methods. Then lamellarity of the liposomes can be determined by using a electron microscopy, then direct microsomal observation, then nuclear magnetic resonance, electron paramagnetic resonance. Then encapsulation efficiency of the product, it can be determined by using a UV visible spectrophotometer or high performance liquid chromatography or the spectrofluorimetry or electron spin resonance spectroscopy, depending on the nature and type of the drug. Then liposome stability can be also evaluated with respect to the physical stability like aggregation, chemical stability, that for example chemical degradation, then the colloidal stability, then degradative damage upon long-term storage by using a HPLC or evaporative light scattering detectors or the MS, that is a mass spectroscopic analysis, lipid peroxidation, that is by using a laser light scattering or by gel filtration columns and freeze fracture electronic microscopy. Then oxidative stabilization also needs to be assessed, and liposome hydrolysis by bichromatic ELISA. So these are the some parameters needs to be evaluated for the liposome stability. Then the characterization assays for the liposomes, there is a basic assay, needs to measure the pH, then the osmolarity by using osmometer, lipid concentrations by using a lipid phosphorus content, HPLC, enzymatic assay, HPTLC, then lipid composition, TLC and HPLC, lipid concentration by lipid phosphorus content, HPLC, enzymatic assay, HPTLC, lipid composition by TLC and HPLC, cholesterol concentration by HPLC, HPTLC, enzymatic assay, lipid hydrocarbon chain composition by GC, residual organic solvents by GC, heavy metals by NMR. The chemical stability, for example, lipid hydrolysis by HPLC and HPTLC, lipid hydrocarbon chain oxidation by lipid peroxidase, conjugated dyes, fatty acid composition, and thiobarbituric acid method. Then free fatty acid concentrations by HPLC and cholesterol oxidation by HPLC and TLC. Then the physical stability, that is appearance, color, clarity by using a pharmacopoeial method. Vesicle size distribution already we have discussed. Micron range already we have discussed. Lamellarity already we have discussed. Zeta potential already we have seen. Then thermotropic phase behavior by DSC, NMR, FTIR, fluorescence spectroscopy or Raman spectroscopy. And phase separation, again differential scanning calorimetry, FTIR, NMR, fluorescence, and so on. Microbiological assay, yes, that is by using the pharmacopoeial methods or the endotoxin, that is a LAL assay. So in this session, we are going to discuss the applications of the liposomes. So let us see the applications of the liposomes. Liposomes are widely used to deliver the various drugs to treat the certain diseases and disorders. Look at the figure. Here I have listed some marketed preparations of the liposomes along with their applications. So the liposomes can be used for the to treat the fungal diseases. It can be used to prepare the certain preparations which are going to act as a analgesics. It can be used for the photodynamic therapy. It can be used in the preparation of the viral vaccines, and furthermore it can be used for the cancer therapy. There are so many preparations are there which are present in the market related with the fungal diseases, analgesics, photodynamic therapy, viral vaccines, and for the cancer therapy. Liposomes are used in the pharmaceuticals to prepare the drug delivery systems. It is used for the diagnostic purpose. It is used in the food science as well as it is used in the cosmetics. The therapeutic areas which is covered by the liposome based products are the cancer therapy, fungal diseases, certain analgesics, photodynamic therapy, and the viral vaccines. Let us see the certain marketed preparations along with their technologies which is used to deliver the drugs. Very first technology which is used to prepare the liposome, it is known as the stealth liposome technology. Here, this is the PEG dilated product. Here, the PEG is used to functionalize the liposomes. The strands of polymers are attached to the drug molecule or the cysteine. There is a covalent linkage of liposome to a PEG protects the active moiety from the recipient immunosystem, which results in a reduced immunogenicity, antigenicity, clearance, and the toxicity. Look at the figure. So here, this is the non-PEG dilated liposome, and this is the PEG dilated liposome. PEG dilated liposome, it is having a long circulation time, isn't it? And slower clearance. Whereas in the case of non-PEG dilated liposomes, they are having the rapid clearance and the less time in a circulation. So, covalent linkage of liposome to a PEG protects the active moiety from the recipient's immunosystem, which results in a reduced immunogenicity, antigenicity, clearance, and the toxicity. One product which is available in the market on the stealth liposome technology known as the Doxil, which is developed by the Swiss Pharmaceuticals. It is given by the intravenous route it is available as an intravenous injection. It contains the active pharmaceutical ingredient that is the doxorubicin. The formulation of the Doxil contains the drug hydrogenated soya phosphatidylcholine, cholesterol, PG2000, diasteroyl SN glycerophosphoethanolamine. It is used for the management of advanced ovarian cancer, multiple myeloma, and HIV associated Kaposi sarcoma. So this is a product that is the Doxil, which is based on still liposome technology. So whatever the liposome is there, it comprises the polyethylene glycol, which protects the active moiety from the recipient immunosystem, which results in reduced immunogenicity, antigenicity, clearance, and the toxicity. So this is about the still liposomal technology used to treat the certain cancers like ovarian cancer, multiple myeloma, and HIV associated Kaposi sarcoma. Let us see non-PEG dilated liposome technology, that is NPL technology. It offers the benefits of PEG dilated liposome while eliminating the side effects associated with PEG, such as a hand-foot syndrome. When we are using the PEG dilated liposomes, it is having a side effects like a hand-foot syndrome, that is HFS. NPL doxorubicin, that is NPLD injection, provides a better safety profile over conventional dox and doxil. NPLD not only reduces the cardiac toxicity associated with the dox, but also the dose limiting toxicity linked with the use of doxil, such as HFS. Our ultimate aim in the treatment of the cancer is to reduce the toxicity of drug to the normal cell. This is achieved by the combination of specific composition and unique manufacturing process of the NPLD liposome, which gives it the desired physicochemical properties. So this is the non-PEG chelated liposome technology. The product which is available in the market, that is known as the Myoset, it is developed by the Elan Pharmaceuticals. It is given by the intravenous route. It contains the doxorubicin as a active pharmaceutical ingredient, and it is formulated by using the egg phosphatidylcholine and the cholesterol. It is used for the metastatic breast cancer in combination with cyclophosphamide. So this is a product that is a Myoset liposomal doxorubicin, right? This is a non-PEG dilated liposome technology which avoids the certain side effects associated with a PEG dilated liposomal systems such as a hand-foot syndrome. Let us see the Depofoam liposome technology. So Depofoam is a extended release drug delivery technology which is developed by the Passira Pharmaceuticals, which encapsulate drug in its multivesicular liposomal platform without modification of their molecular structure. The multivesicular liposome releases the drug ranging from 1 to 30 days. The Depofoam consists of the microscopic spheroids. So these are the microscopic spheroids which is having a size 3 to 30 micrometer with a granular structure and single layered lipid particle composed of honeycomb of numerous concentric internal aqueous chamber containing the bounded drug. There are certain products which are available in the market on the technology, that is a Depofoam liposome. The product that is the Deposite, that is cytarabine liposome injection developed by the Sky Pharma. It is given by the spinal root. It contains the API cytarabine and formulation comprises the cholesterol, trilene, palmitoyl phosphatidylcholine, dipalmitoyl phosphatidylglycerol, and it is used for the lymphomatous meningitis. So this is a product that is a Deposite, which is given by the spinal root. Yet another product is available in the market, that is the Depodur. It is an extended release liposome injection, which is given by the epidural route. It contains the drug that is a morphine sulfate. It comprises the cholesterol, tryline, diollyl phosphatidylcholine, and dipalmitoyl phosphatidylglycerol. It is used for the pain management following major surgery. And this is a product which is based on the Dipofoam liposomal technology, that is a Dipodurfoam. So the Xparal, it is a bupivacaine liposome injection suspension, which is developed by the Passira Pharma, which contains the API bupivacaine. It is given by the subcutaneous route, and it is used for the post-surgical local analgesia and as an intercellular branchial plexus nerve block to produce the post-surgical regional analgesia. So this is a product that is a Xparal. Yet another product that is a Daunosome, which contains the daunorubicin. It is a daunorubicin citrate liposome injection developed by the Nexter Pharma. It is given by the intravenous route, and it is formulated by using the cholesterol and diasteryl phosphatidylcholine, and it is used for the AIDS related Kaposi sarcoma. Few other products of the liposome which are available in the market, that is the first one, that is Mepact, which is developed by the Takeda Pharma, which contains the drug mefamutide. It is given by the intravenous route. Formulation comprises diollyl phosphatidylserine, palmitoyl phosphatidylcholine, and it is used for the high grade resectable non-metastatic osteosarcoma. So this is a product that is the Mepact, which is developed by the Takeda Pharma. Then the Morcubo, which is developed by the Talon Therapeutics, which comprises the drug vincristine. It is given by the intravenous route, and it is formulated with the sphingomyelin and the cholesterol, and it is used for acute lymphoblastic leukemia. So this is a product that is a Morcubo vincristine sulfate liposome injection given by the intravenous route as an intravenous infusion. Then Onivide, it is yet another liposomal based product which is developed by the Merrimack Pharma, which contains the drug irinotecan. It is given by the intravenous route. The formulation comprises the diasteryl phosphatidylcholine, methoxypropylene glycol 2000, stearoyl SN glycerophosphoethanolamide. It is used in a combination therapy with the fluorouracil and leucovorin in metastatic adenocarcinoma of pancreas. So this is a product Onivide. Yet another product is there, that is a Abelset, which is developed by the Sigma Tau Pharma, which comprises the drug amphotericin B. It is given by the intravenous route, and it is formulated by using the dimyristyl phosphatidylcholine and diasteryl phosphatidylglycerol. It is used for invasive severe fungal infections. Then Ambisome, it is developed by the Astellas Pharma. Again, it contains the amphotericin B to treat certain fungal infections given by the intravenous route, and it contains the cholesterol, hydrogenated soya phosphatidylcholine, and diasteryl phosphatidylglycerol. So this is a product Ambisome. Then Ampotec, it is developed by the Benvenu lab. It contains the amphotericin B. It is given by the intravenous route, formulated with the cholesterol sulfate, and it is used for severe fungal infection. Then the Visudyne, it is developed by the Novartis. It comprises the drug vertiforfin given by the intravenous route, and formulation comprises dimyristoyl phosphatidylcholine and 8-phosphatidylcholine. It is used for the choroidal neovascularization, and this is a product that is the Visudyne. Then the another product that is Ifaxal, which is developed by the Crucial. It contains the API inactivated hepatitis A virus given by the intramuscular route, formulated with the dioleal SN glycerophosphoethanolamine and palmitoyl phosphatidylcholine, and it is the vaccine for the hepatitis A, and this is a product that is the Ipaxal. Then the another product that is a Inflexal V. It is developed by the Crosal. It is the inactivated hematoglutinin of influenza virus strain A and B. It is a vaccine given by the intramuscular route. It is formulated with the dioleal SN glycerophosphoethanolamine and palmitoyl phosphatidylcholine, and it is used for the influenza. So this is a product Inflexal developed by the Crosal. Yet another product is there, that is Aricaceae, which is developed by the Insmed, which contains the API that is amikacin. It is given by the pulmonary route by using the device eFlow. Formulation comprises the dipalmitoyl phosphatidylglycerol and the cholesterol, and it is used for Mycobacterium avium complex lung diseases. So this is a product that is Aricaceae. Then lipoplatin, which is developed by the Regulon. It contains the drug cisplatin. It is given by the intravenous route. Formulation contains DPGG, SOAPC, MPEG, DSP, lipid conjugate, and the cholesterol, and it is used for the pancreatic cancer. So this is a product lipoplatin, which is developed by the Regulon. Then Mosquirix, which is developed by the GSK. It comprises the RTSS, that is the portion of P. falciparum circumsporozoid protein fused with hepatitis B surface antigen, that is RTS, and combined with hepatitis B surface antigen. It is used for Plasmodium falciparum and hepatitis B vaccine. So this is a product that is a Mosquirix. Yet another technology which is available in the liposome, that is the lysolipid thermally sensitive liposome technology, that is LTSL technology. This is a novel liposomes exhibit temperature dependent release of encapsulated drugs. So this is a technology which utilizes the temperature as a stimulus and releases the drug. Local tissue temperature, it is generally elevated to 42 degrees Celsius by the radio frequency ablation. So here we can utilize the radio frequency to increase the temperature of the tissue. Lipid components present in the liposome undergo a gel to liquid transition at elevated temperature and making it more permeable and thus releasing the drug at a specific site. Moreover, application of local hyperthermia causes the leakage of blood vessels within the tumor and thereby increasing the accumulation of liposomes in the tumor. So look at the figure, how it is going to be work. So there is a product which is available or technology, that is Thermodox, which is developed by the Celson Corporation, uses the LTSL, that is lysolipid thermally sensitive liposome technology, to encapsulate the doxorubicin for the treatment of various solid tumors. So we have to supposed to give the product by parenteral route, then at a specific tissue, on the specific tissue we have to supposed to apply the radio frequency. If there is a liposomes which are reached over there due to thermal ablation and the change in the temperature, the liposome releases the drug at a target site or the specific site. So this is about the lysolipid thermally sensitive liposome, the product which is available in the market, which is based on the thermodox technology, which comprises the drug doxorubicin for treatment of the solid tumors. Let us see some benefits of liposomes, which is specific to the drugs. So the first one, that is the improved solubility of lipophilic and amphophilic drugs. So here I have listed some drugs. In these cases, the solubility of drug, it is going to be increased. That is amphotericin B, porphyrins, minoxidil, some peptides, anthracyclines, hydrophilic drugs such as anti-cancer agent like doxorubicin and acyclovir. Then passive targeting to cells of immune system, especially cells of a mononuclear phagocytic system. For example, antimonials, amphotericin B, then porphyrins, vaccines, and immunomodulators. Then sustained release system of systemically or locally administered liposome. For example, doxorubicin, cytosine arabinoside, corticosteroids, biological proteins, peptides such as vasopressin. Site avoidance mechanism, for example, doxorubicin and amphotericin B. Site-specific targeting, anti-inflammatory drug, anti-cancer, and anti-infectives. Improved transfer of hydrophilic and charged molecules, for example, antibiotic, chelators, plasmids, and genes. And improved penetration into tissue, for example, corticosteroid, anesthetics, and the insulin. So these are the applications of the liposome. Let us see the summary. So in the liposomes, we have discussed the introduction to liposomes, definition, structure, then the advantages and disadvantages of liposome. Furthermore, we have discussed the types of liposome, then the properties and mechanism of action of liposomes, then components of liposomes, methods of preparation of liposomes, and finally the applications of liposomes along with the some advancements in the liposomal technology to deliver the drugs. Let us see the references. So these are the few references which I have referred for the presentation, that is liposome and their applications in cancer therapy, liposomes as a novel drug delivery system, fundamental and pharmaceutical applications, liposome formulations in clinical use and updated review, liposomes a review of manufacturing technique and targeting strategy, liposome classification, preparation, and the applications. Thank you very much. Hope all of you have understood the concepts related with the liposomes.